Hello everybody, welcome back to my channel. In this video, I have collected some important points from the chapter The Fundamental Unit of Life. You could use these points to prepare for your exams. Let's get into the video. Discovery of cell. Cells were first discovered by Robert Hooke in the year 1665. Living cells were discovered by Leeuwenhoek in 1674. Robert Brown discovered the nucleus of the cell in the year 1831. Purkinje coined the term protoplasm for the fluid substance of the cell in the year 1839. Virchow expanded the cell theory by suggesting that all cells arise from pre-existing cells in the year 1855. Cell theory. All the plants and animals are composed of cells and that the cell is the basic unit of life. Cell theory was presented by two biologists, Sheldon and Schwann. Facts on cell. Cell in Latin means little room. Cell is a basic unit of life. The shape and size of the cells are related to the function they perform. Each cell has got a certain specific component within it known as cell organelles. Organisms. Unicellular organisms, multicellular organisms. Unicellular are organisms with single cell and multicellular are organisms with many cells. Examples of unicellular organisms are bacteria and paramecium. And the examples of multicellular are plants and animals. What is cell made up of? The three features are almost there in every cell. They are plasma membrane, nucleus, and cytoplasm. Plasma membrane is flexible and is made up of organic molecules called lipids and proteins. The flexibility of the cell membrane also enables the cell to engulf in food and other material from its external environment. Such processes are known as endocytosis. Amoeba acquires its food through such processes. Diffusion. Spontaneous movement of substance from a region of high concentration to a region of low concentration. Osmosis. Movement of water molecules to selectively permeable membrane from a region of high water concentration to a region of low water concentration is called osmosis. What will happen if we put an animal cell or a plant cell into solution of sugar or salt water? Hypertonic solution. A solution that has low solute concentration than the one to which it's compared. A cell placed in it will gain water and is likely to swell up. Isotonic solution. A solution that has the same density as another solution with which it is compared. A cell placed in it will neither gain or lose water. Hypertonic solution. A solution that has high solute concentration than the one to which it is compared. A cell placed in it will lose water and shrink. Cell wall. Plant cells, in addition to the plasma membrane, have another rigid outer covering called the cell wall. The cell wall lies outside the plasma membrane. The plant cell wall is mainly composed of cellulose. Cellulose is a complex substance and provides structural strength to plants. Plasmolysis. When a living plant cell loses water through osmosis, there is shrinkage or contraction of the contents of the cell away from the cell wall. This phenomenon is known as plasmolysis. Nucleus. Nucleus is a dot-like structure in the center of each cell. It has a double-layered covering called nuclear membrane. The nucleus contains chromosomes. Chromosomes are composed of DNA and protein. An undefined nuclear region containing only nucleic acid is called nucleoid. Now let's see the difference between prokaryotes and eukaryotes. Organisms whose cell lack a nuclear membrane are called prokaryotes. Membrane-bound organelles are absent. The nuclear region contains only nucleic acid and is undefined due to the absence of nuclear membrane and is known as nucleoid. Eukaryotes. Organisms with cell having nuclear membrane are called eukaryotes. Membrane bound organelles are present. The nuclear region is well defined and surrounded by a nuclear membrane. Cytoplasm. The cytoplasm is a fluid contained inside the plasma membrane. It also contains many specialized cell organelles. Each of these organelles perform a specific function for the cell. Cell organelles. Each cell has got certain specific components within it, known as cell organelles. Each kind of cell organelle performs a specific function. Some important examples of cell organelles are endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, and plastids. Endoplasmic reticulum. There are two types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum. It looks rough under the microscope because it has particles called ribosomes attached to its surface and are the sites of protein manufacture. Smooth endoplasmic reticulum looks smooth under the microscope and helps in the manufacturing of lipids. Membrane biogenesis. Some of these proteins and lipids help in building the cell membrane. This process is known as membrane biogenesis. Golgi apparatus. It was first described by Camille Golgi. It consists of a system of membrane bound vesicles arranged approximately parallel to each other in sacs called cisterns. Functions. It carries out the storage, modification, and packaging of substance manufactured in the cell and is also involved in lysosomes formation. In some cases, complex sugars may be made from simple sugars. Lysosomes. Lysosomes are membrane-bound sacs filled with digestive enzymes. They are waste dispersal system of the cell. They keep the cell clean by digesting any foreign material as well as worn-out cell organelles. When the cell gets damaged, lysosomes may burst and the enzymes will digest their own cell. Therefore, lysosomes are also known as the suicide bags. Plastids. Plastids are present only in plant cells. Like the mitochondria, plastids also have their own DNA and ribosomes. There are two types of plastids: chromoplasts and leucoplasts. Chromoplasts. Chloroplasts containing the pigment chlorophyll are known as chloroplasts. Chloroplasts are important for photosynthesis in plants. They also contain various yellow or orange pigments in addition to chlorophyll. The internal organization of the chloroplast consists of numerous membrane layers embedded in a material called stoma. Leucoplasts. Leucoplasts are primarily organelles in which materials such as starch, oils, and proteins granules are stored. Vacuoles. Vacuoles are storage sacs for solid or liquid contents. Vacuoles are small size in animal cells, while plant cells have huge vacuoles. The central vacuole of some plant cells may occupy 50 to 90% of the cell volume. In plant cells, vacuoles are full of cell sap and provide turgidity and rigidity to the cell. In single-celled organisms like amoeba, the food vacuole contains the food item that the amoeba has consumed. Mitochondria. Mitochondria are known as the powerhouse of the cell. Mitochondria have two membrane coverings. The outer membrane is porous and the inner membrane is deeply folded. The energy required for various chemical activities needed for life is released by mitochondria in the form of ATP molecules. ATP is known as the energy currency of the cell. Mitochondria have their own DNA and ribosomes and able to make their own proteins. Cell division. The process by which new cells are made is called cell division. There are two types of cell division: mitosis and meiosis. Mitosis. This is the process by which most of the cells divide for growth. It is necessary for growth and repair of tissues in organisms. Mother cell divides to form two identical daughter cells. The daughter cells have the same number of chromosomes as mother cells. Meiosis. This process involves two consecutive divisions. It is necessary for sexual reproduction. Mother cell divides to form 4 new cells instead of just 2. The new cells only have half the number of chromosomes than that of the mother cell. Thank you for watching. Hope this video helped you. Please like, share and subscribe. See you in the next video. Bye.